Welcome students. Today we shall discuss regarding remaining cell organelles. Previously we had discussed chloroplast, mitochondria, Golgi body, endoplasmic reticulum, and now today we are going to discuss ribosome, centrosome, peroxisome, vacuoles, and prokaryotic and eukaryotic cell. If we talk about ribosomes are the granular structures present freely in the cytoplasm, or they can be found adhered to endoplasmic reticulum both in plant and animal cell. These ribosomes are responsible for the synthesis of protein for the cell. It is the only cell organelle which lacks any membrane around it. Here you can see the diagram. You find here it is written here 80S and here it is 70S. Ribosome in fact is the organelle which is made up of two different units. This one is the smaller unit, this one is the larger unit, and these two units sometimes remain separated or sometimes remain attached. In case of prokaryotic and eukaryotic cell, although it is going to be discussed later on, but in your previous class also you must have read about prokaryotic and eukaryotic cell. These prokaryotic cell and eukaryotic cell have different kinds of ribosome. Here you can see that eukaryotic cell has 80S ribosome, whereas the prokaryotic cell has 70S ribosome. And both the subunits of ribosomes, in case of 80S ribosome, the smaller subunit is of 40S and the larger subunit is of 60S, whereas in prokaryotic cell, the 70S ribosome is found, which has the smaller 30S subunit and larger 50S subunit. This is all about ribosome, but one very important question arises here, that we organisms through our nutrition take protein. Protein is taken by every organism during the course of its nutrition. Even then, why is it so that cell require to form protein with the help of ribosome? This is a very important question. I think I must give you the answer. It is not the live class. Had it been there, I would have asked you people regarding this question. In fact, our body requires a large number of enzymes and proteins for different metabolic functions. If we take protein through our nutrition, this protein is digested in our body and amino acids are released. These amino acids are supplied to the cell. Thereafter, cell has to decide what kind of enzyme it has to produce, and with the help of those amino acids that our cell has obtained through nutrition, that amino acid is again used by these ribosomes to make different kinds of proteins or enzymes. The next one is the centrosome. Centrosome is one other important organelle that lies near the nucleus of the cell. It is an organelle usually located near the nucleus of the cell. It is supposed to play an important role at the time of cell division. It is constituted of two centrioles. You can see here these are the two centrioles present inside the centrosome. At the time of cell division, centrosome divides and each centriole moves to opposite direction and help in the formation of spindle fiber at the time of cell division. Here is the diagram of a cell where it is under the process of division. You can see here that the centrioles has moved to opposite poles and chromosomes have arranged at the equatorial plate. You can see here chromosomes at metaphase stage of cell division. Metaphase is the stage of cell division. At this stage, chromosomes are found to lie arranged at the equatorial plate of the cell, and the spindle fibers get attached with the centrosome of the chromosome. So these spindle fibers are formed with the help of these centrioles, and these spindle fibers help in the process of cell division. Okay. Thereafter, The another cell organelle is the peroxisome, like lysosome. Peroxisomes are membrane-bound cell organelles. They are usually found in the cells of liver and kidney and are supposed to detoxify the cell. Vacuole is one another organelle inside the cell. These are the large spaces inside the cell, usually larger in plant cell and smaller in animal cell. Various types of vacuoles are there. One is the food vacuole, the other one is the contractile vacuole. Food vacuole is the vacuole that is responsible for the digestion of food. You must have seen in case of amoeba. Most of the aquatic organisms which remain in water, sometimes lot of water enter inside the cell and this water is unnecessarily present there inside the cell. So cell automatically removes this water with the help of this contractile vacuole and the process is known as osmoregulation. That is regulation of water requirement by the cell. This is all about the different cell organelles. We have discussed almost every cell organelle of the cell, and now the other topic that we require to discuss, that we need to discuss, is the prokaryotic and eukaryotic cell. In fact, cells are basically of two types as far as the nature of the cell is concerned. One is called the prokaryotic cell and the other one is called the eukaryotic cell. And we can identify or we can distinguish these two different kinds of cell on the basis of these following features. Here, in case of prokaryotic cell, it has no organized nucleus. It means the nucleus inside the prokaryotic cell has no nuclear membrane, whereas the eukaryotic cell has organized nucleus. The other important feature is the prokaryotic cell has no membrane bound organelles. Every organelle that we have discussed earlier, if we see in case of prokaryotic cell, we don't find such organelles which have membrane around it. So the prokaryotic cell has no membrane bound cell organelle, whereas the eukaryotic cells have membrane bound organelle. In case of prokaryotic cell, cell size is little smaller. In case of eukaryotic cell, the cell size is little larger. The ribosome we have seen earlier. In case of prokaryotic cell, the ribosomes are 70S, whereas in case of eukaryotic cell, these ribosomes are 80S. And the another important feature of prokaryotic cell is that the DNA in prokaryotic cell has no protein associated with it, whereas in eukaryotic cell, DNA is associated with protein. That's all. Thank you. If we any inquiry, you can ask me on my mobile number 7050773907. Thank you.